Imagine sitting courtside at a professional basketball game. The arena around you is just completely deafening.
Oh, yeah, it's absolute sensory overload.
Right. And you are watching a fast break unfold in real time. You've got 10 bodies moving at sprinting speeds across the hardwood.
Sweat flying, sneakers squeaking,
Just the sheer physical mass of these players colliding. And you see an elite point guard driving toward the basket, absolutely surrounded by towering defenders.
Defenders who are, actively trying to disrupt every single spatial pathway available.
Exactly. And then in a fraction of a second, without even turning their head, that player executes a flawless, pinpoint, no-look pass to a teammate who is cutting through the chaos for an easy layup.
It looks like magic.
It really does. It looks like they have eyes in the back of their head. And we are so conditioned to marvel at the physical attributes of athletes, right? Their height, their wingspan, their vertical leap.
Sure, the raw physical hardware.
But when you look at that no-look pass and you slow it down to the millisecond, you really have to ask, is what we just witnessed primarily a physical achievement, or is it actually a neurological masterpiece?
That is, that underlying tension between the physical and the neurological is really the frontier of modern sports science.
Right.
Because that no-look pass is fundamentally a triumph of information processing. It requires a brain that can seamlessly track multiple moving targets simultaneously.
While running at top speed.
Exactly.
Yeah.
Calculating trajectory and force in a three-dimensional space, anticipating the future positions of both teammates and opponents based on, these tiny micro-movements.
And doing all of that while actively inhibiting the primal urge to react to the massive sensory distractions happening all around them.
It's staggering when you really break it down.
So that is the mission for this deep dive. We are going to explore the psychology and the neuroscience of athletic expertise, specifically zooming in on basketball.
It's such a perfect sport for this.
It is. We are going to completely unpack how the brains of expert athletes physically and functionally differ from those of amateurs. And we're going to look at what scientific techniques actually work to build an elite athlete's brain.
And we are relying on a very specific stack of peer-reviewed scientific papers to back up the claims we make today.
No guessing here.
Right. We want to get into the actual biological tests, the effect sizes, and the experimental conditions that neuroscientists use to map the expert brain.
And if you are listening to this, I really want to emphasize that whether you're an athlete yourself trying to improve your physical performance, or, someone trying to optimize how you learn complex skills at work.
Or even if you just have a deep curiosity about the absolute limits of human potential.
Exactly. This exploration is going to completely reframe how you view the concept of talent. We are diving deep into neuroplasticity, the brain's literal ability to reorganize its physical architecture by forming new neural connections.
It's going to be a wild ride.
I think the best place to start is at the sensory level, because before we can even look at how an athlete moves their body, we have to look at how they perceive the world around them.
Right. Perception always precedes action.
So I've read a bit about the cognitive psychology of visual attention, and the classical model is always described as a spotlight.
Ah, the spotlight theory. Yes.
You have this beam of attention that sweeps across your visual field. You focus on the ball, then you shift the spotlight to the defender, and you shift it to the hoop.
Which makes sense intuitively, right?
It does, but a point guard clearly can't just be sweeping a single spotlight around when 10 bodies are moving at once. It would just be way too slow.
Yeah, the spotlight theory really dominated cognitive psychology for decades. It basically assumed a single focus of selection, almost like a second fovea that roams around your visual field independently of your eye movements.
Like a flashlight in a dark room.
Right. But many everyday activities, and certainly highly dynamic sports like basketball, require us to pay attention to multiple regions of interest simultaneously.
So how do we actually do that?
Well, to understand that, there is a foundational paper from 2005 in the journal Trends in Cognitive Sciences. It's by Patrick Cavanagh and George A. Alvarez, titled Tracking Multiple Targets with Multifocal Attention.
Okay, multifocal attention.
Yeah. They highlight a pivotal shift in the field that started with Zenon Polishin's work in the late 1980s. That work completely overturned the single spotlight view using something called the multiple object tracking task.
Let's break that down. If I were a subject sitting in a cognitive psychology lab doing a multiple object tracking task, what am I looking at on the screen? Give me the mechanics.
Okay, so you are looking at a computer screen displaying an array of identical items, let's say 10 simple black disks.
Just dots on a screen.
Yeah, and they are moving randomly within a rectangular area, constantly bouncing off the invisible borders of the screen and bouncing off each other.
Like a chaotic game of billiards or, a DVD logo bouncing around.
Exactly like that. Now, before the movement begins, a subset of those disks, maybe 4 of them, are briefly highlighted in red. Those are your assigned targets. A second later, they turn back to black, becoming visually identical to all the distractors on the screen, and the movement begins.
Oh, wow. So you have to track them while they look exactly like the ones you're trying to ignore.
Yes. They move completely randomly for about 7 to 15 seconds. When they finally freeze, one random disk is highlighted, and you have to report. Was that one of your original targets, or was it a distractor?
That sounds incredibly taxing.
Mm.
I mean, if they all look completely identical and they are constantly crossing paths, I would imagine the human limit for tracking independent moving targets is pretty low.
You'd think so. But the research consistently shows that the normal human capacity is tracking about 4 independent targets.
4? Really?
Yeah, most subjects can keep track of 4, sometimes 5 targets over several seconds with high accuracy.
That's higher than I expected.
What makes this so profound is that subjects cannot use eye movements alone to follow them. The targets are moving too unpredictably and too far apart.
Right, you can't just fixate your eyes on one and hope to track the rest peripherally.
Nor can a single spotlight of attention just widen to encompass all the targets. Because if you widen the spotlight, you are inherently picking up all the distracting disks mixed in among them.
Which completely ruins the tracking.
Exactly.
So the single spotlight theory falls apart. If my eyes are fixed on the center of the screen, but I am mentally tracking 4 distinct black disks bouncing around, how is my visual cortex managing that?
Cavanagh and Alvarez outline 2 prominent models that attempt to explain the underlying mechanism. The first is the multifocal attention model, and the second is Polition's finest model, which stands for fingers of instantiation.
Figures of instantiation. Oh.
Yeah. The multifocal attention model is fairly straightforward in its premise. It assumes that the brain can literally split its attention into separate, independent foci.
So abandoning the single spotlight entirely and replacing it with multiple independent tracking lasers.
That is a brilliant way to visualize it, yes. Multiple tracking lasers.
And what about the NSK model?
The finest model is slightly different. It proposes that the brain assigns an index, or like a preconceptual pointer, to a target in the early stages of visual processing.
Like putting a mental sticky note on it.
Exactly. Once that index is attached, it sticks to the target as it moves, without needing constant active refreshing from a central executive attentional resource.
So it's sort of running in the background.
Yeah, it acts like a sticky tracker that allows your higher level attention rapid access to the object whenever it needs to query its location. So the underlying indexing is distributed, even if the conscious attention periodically polling those indices is singular.
That makes a lot of sense. But there was a detail in the Cavanagh and Alvarez paper about the biological constraint of this tracking capacity. It's not just a general cognitive limit of 4 objects anywhere. It's tied to the actual physical geography of the visual cortex, right? The hemifield split.
Yes. This is perhaps the most fascinating physiological constraint they discuss. The capacity to track targets is entirely hemifield independent.
Okay, explain hemifield independent for us.
As you might know, human vision is processed contralaterally. The left side of your visual space is processed by the right hemisphere of your visual cortex, and the right side of your visual space is processed by your left hemisphere.
Right, the wiring crosses over.
Exactly. The research demonstrates that if your total tracking capacity is 4 items, you do not have a general floating pool of 4 trackers to deploy wherever you want.
Oh, interesting.
You have a strict limit of 2 trackers isolated to your left visual field and 2 trackers isolated to your right visual field.
Wait, I really want to make sure I understand the implication that if I have 3 targets moving around entirely on the left side of my vision and only 1 target on the right side, I will struggle or fail to track that third one on the left, even though my brain's total capacity is 4.
Yes, absolutely. If the experiment forces the targets to remain within their respective hemifields, tracking 2 on the left and 2 on the right is well within normal human limits.
But 3 on one side breaks the system.
Trying to track 4 items in a single hemifield results in massive performance degradation. Wow.
This hemifield independence strongly suggests that the early stages of attentive tracking are deeply rooted in the retinotopic cortical areas of the brain. the early visual areas like V1, V2, and V3.
Where the physical layout of the neurons still rigidly maps to the physical layout of the retina.
Exactly. Your cognitive limits are dictated by the anatomical boundaries of your hemispheres.
That is a wild constraint for an athlete. I mean, a basketball court isn't going to politely organize the opposing team so that 2 players stay in your left peripheral vision and 2 stay in your right.
No, they certainly are.
So if an elite point guard is managing to track the chaotic movement of 5 or 6 players on one side of the court, they must be pushing past this baseline biological limit. Can this specific multifocal tracking capacity be trained, or is it a fixed genetic ceiling?
It is highly trainable. Cavanagh and Alvarez note that certain professions demanding high multiple target tracking, like air traffic controllers or radar operators, show higher baseline tracking capacities than the average undergraduate student.
That makes sense. Lots of practice. But they also cite incredibly compelling research by Green and Bavelier regarding action video games.
Video games? Really?
Yeah. They found that playing fast-paced, first-person action video games significantly alters and improves visual attention abilities. And the neuroplastic response is shockingly fast.
How fast are we talking?
Just 10 hours of focused training on an action video game resulted in measurable improvements in visual attention scores.
10 hours. 10 hours to alter a fundamental cortical processing limit. What exactly is improving there? Are they just getting faster at moving their eyes?
It's much deeper than ocular motor speed. The gamers developed the ability to process visual information faster. They could apprehend more items at a single glance, and crucially, they developed smaller zones of interaction.
Okay, what is a zone of interaction?
Think of the attentional pointer attached to a moving target not as a single pixel, but as a small halo of cognitive focus.
Like a glowing bubble around the player you're tracking?
Right. If a distracting object enters that halo, that zone of interaction, the brain struggles to maintain the identity of the target, and they often get swapped or lost.
Oh, I see.
Gamers, and by extension elite athletes who train their visual fields, manage to shrink those halos. Their attentional resolution becomes sharper.
So the bubble gets tighter.
Yes. So when a target and a distractor pass extremely close to one another, like a teammate running a tight screen off a defender, the highly trained visual cortex is less likely to suffer from interference and drop the tracking index.
That is fascinating. So the perceptual foundation of the elite athlete is a highly refined, high-resolution, multifocal tracking system.
Right.
But tracking only gets you so far, right?
Right.
If you are just tracking where everyone is at this exact millisecond, you are still reacting to the present. And I've always heard that the defining characteristic of a professional athlete isn't their reaction time, it's their anticipation.
Which brings us perfectly to the evolution from perception to action. To explore that, we turn to a really comprehensive review paper published in Nature Review's Neuroscience in 2009.
Okay.
It's by Yarrow, Peter Brown, and John W. Krakauer. The title is Inside the Brain of an Elite Athlete: The Neural Processes That Support High Achievement in Sports.
A very fitting title.
Yarrow and his colleagues synthesized decades of research to show that the hallmark of athletic expertise is the shift from reactive decision-making to predictive decision-making.
Because basic human reaction time have a hard biological floor, doesn't it? I mean, if I see a stimulus and have to execute a motor response, the neural signal has to travel from my retina to my visual cortex to my motor cortex, down my spinal cord to my muscles.
It's a long physical journey for an electrical signal.
That physical transit time has to take hundreds of milliseconds.
It does. A simple auditory reaction time might be around 150 milliseconds, and a visual reaction time is closer to 200 or 250 milliseconds for an average person.
And in elite sports, a quarter of a second is an eternity.
Right. In elite sports, reacting to a visual cue after it has fully materialized is catastrophically slow. If a tennis player waits to see the ball leave the opponent's racket before deciding where to move, or if a basketball defender waits to see the ball fully released from the shooter's hand before jumping, they have already lost. The event is over.
So how are they acting on the future? Is it just like pattern recognition?
It is a deeply ingrained, highly complex form of pattern recognition born out of what K.A. Erickson termed deliberate practice in his seminal 1993 work.
Right, the 10,000 hours concept.
Exactly. Yarrow's paper details how experts develop incredibly sophisticated internal models of their specific sport. They extract advanced kinematic cues from their opponents long before the primary action occurs.
Advanced kinematic cues. So what does that look like on the court?
Well, a novice watches the ball, an expert watches the angle of the opponent's shoulder, the rotational velocity of their hips, the subtle shifting of weight on their plant foot. They use these micro movements to computationally anticipate an action before it is executed. They are fundamentally acting on a predicted future state, not reacting to a past state.
I'm trying to visualize the metabolic cost of this.
Okay.
If an athlete's brain is simultaneously maintaining high-resolution multifocal tracking on multiple targets, while continuously running complex kinematic predictions on the micro-movements of an opponent's hips and shoulders, while also preparing a coordinated motor response,
It sounds exhausting.
it seems like their brain should be redlining. If we put an elite athlete in a scanner while they are performing these complex sports-specific tasks, I would expect their cortex to be burning massive amounts of glucose, just lighting up with widespread activation compared to a beginner who's barely processing the basics.
It is the most intuitive assumption to make. We generally equate intense cognitive or physical output with increased neural processing effort.
Right. Heavy lifting for the brain.
If a task is incredibly complex, the brain must be working incredibly hard to execute it. But the neuroimaging data reveals a completely inverted reality.
Really?
Yes. This brings us to a highly counterintuitive concept known as the neural efficiency hypothesis.
The neural efficiency hypothesis. So you're saying the expert brain is somehow doing more processing while extending less energy.
Exactly.
How do neuroscientists even test that without running into massive confounding variables? Because if you just compare my brain trying to play basketball against LeBron James's brain, you're not just measuring basketball expertise, you're measuring a million different variables.
Oh, absolutely. Different baseline intelligence, cardiovascular fitness, anxiety.
Different levels of baseline effort. It's an apples to oranges comparison.
Controlling for those variables is the hardest part of sports neuroscience. To see how it is done properly, we can examine a highly elegant study published in Frontiers in Psychology in December 2019.
Okay, let's hear it.
It's by Lanlin Zhang and a team of researchers titled Neural Efficiency and Acquired Motor Skills, an fMRI study of expert athletes. Zhang's team recognized the exact problem you just described.
The apples to oranges problem.
Yes. Previous studies that compared experts directly to novices yielded conflicting results. Some showed increased brain activation, some showed decreased activation, precisely because individual human differences muddied the data.
So how did Zhang isolate the specific variable of sports expertise?
They utilized a factorial fMRI design with a brilliant control mechanism. They didn't recruit novices at all.
Right.
They recruited two distinct groups of highly trained elite athletes, professional basketball players and professional volleyball players.
Oh, that is incredibly smart.
Yeah.
By doing that, they immediately control for general athletic fitness, competitive mindset, spatial awareness, all the traits that elite athletes share regardless of their specific sport.
Exactly. The physiological baseline is leveled. The experimental design required both groups to perform a motor imagery task while lying inside an fMRI scanner.
Just imagining the movement.
Right. They were instructed to vividly imagine themselves performing two specific actions, throwing a basketball free throw and serving a volleyball.
Now, I know that in an fMRI, functional magnetic resonance imaging, the subject has to remain absolutely still because the machine is highly sensitive to motion artifacts.
Very sensitive, yes.
How reliable is a motor imagery task for mapping actual physical sports skills? Does imagining a movement really engage the motor cortex in the same way as actually doing it?
It is an established and highly robust paradigm in neuroscience. While the primary motor cortex, the area that actually sends the final electrical impulse down the spinal cord to the muscle, might show attenuated activity to prevent you from actually moving in the scanner.
Right, so you don't punch the machine.
Exactly. But the supplementary motor areas, the premotor cortex and the parietal regions involved in planning, sequencing, and spatial mapping, they light up almost identically.
That's fascinating.
The brain prepares the entire blueprint for the movement, it just inhibits the final execution signal.
So the experimental design is a cross-validation setup. The basketball players imagine the basketball throw, which is their domain of expertise, and they imagine the volleyball serve, which is the novice condition for them.
Yes.
And the volleyball players do the exact same thing in reverse. The volleyball serve acts in the internal control condition for the basketball players.
You've summarized the factorial design perfectly. Inside the fMRI, the researchers measured the bold signals, the blood oxygen level dependent signals.
Let's quickly define the physics of the bold signal for the listener, just so we are clear on what the fMRI is actually measuring. It's not measuring the electrical firing of neurons directly, is it?
No, fMRI has poor temporal resolution for measuring instant electrical spikes, but incredible spatial resolution. It relies on hemodynamics blood flow.
Okay, blood flow.
When a localized cluster of neurons fires rapidly, it consumes the local supply of oxygen. In response, the brain's vascular system quickly dilates capillaries in that specific region to deliver a surge of oxygen-rich blood to replenish it.
Like sending supplies to the front lines.
Precisely. And oxygenated hemoglobin has different magnetic properties than deoxygenated hemoglobin.
Oh, because of the iron in the blood.
Yes. The massive magnetic field of the fMRI scanner detects that tiny magnetic difference, allowing researchers to map exactly which millimeter-sized cubes of the brain, called voxels, are demanding the most blood and therefore are the most active.
And in this study, Zhang's team was specifically looking at the bold E signals in the parietal premotor circuit, because that is the network responsible for motor planning and spatial coordination.
Right.
What did the fMRI data reveal when the basketball players imagined their own highly practiced free throw versus the unfamiliar volleyball serve?
The results were a textbook demonstration of neural efficiency. The researchers found a strong negative correlation between behavioral performance or expertise and functional neural measures.
Negative correlation.
Yes. When the elite basketball players imagined their own sport, they showed significantly less intense and much more spatially restricted brain activity in that parietal premotor circuit compared to when they imagined the volleyball serve.
Wow.
And the exact inverse was true for the volleyball players. Their brains worked much harder to imagine the basketball through.
Here's where it gets really interesting. So, true mastery isn't about maximum effort. It's about maximum optimization. When I am doing something I am a true master at, my brain is literally working less hard, consuming less oxygen, and recruiting fewer neurons than when I am fumbling through a task I am an amateur at.
Beautifully said. The expert brain is quieter.
It's actually chilling out while performing the task.
It is quieter because it is profoundly optimized. Zhang's paper contextualizes this using theories of automaticity, specifically referencing work by Logan and Crump from 2009.
How does that work biologically?
When you first learn a complex motor skill, your brain lacks a dedicated pathway for it. So it recruits massive, widespread, distributed networks across the cortex.
Like trying to find your way through a new city without a map.
Exactly. You are consciously utilizing your prefrontal cortex to remember the rules, your visual cortex to track your limbs, your parietal lobe to manage spatial awareness. It is a highly demanding, conscious, step-by-step processing loop.
But over thousands of hours of deliberate practice, what physically changes in that network?
The brain undergoes massive synaptic pruning and consolidation. It identifies the most efficient neural pathway to execute that specific motor command and heavily myelinates those connections, speeding up the transmission.
Paving the dirt road into a superhighway.
Yes. And concurrently, it actively prunes away non-essential inputs and task-irrelevant sensory processing. The skill shifts from conscious, step-by-step control to an encapsulated automatic subroutine.
So the brain no longer needs to recruit the massive distributed network. It just triggers the highly localized, incredibly efficient automated pathway.
Exactly.
I would imagine that this biological shift toward automaticity explains a lot of sports psychology phenomena.
Absolutely.
If a skill becomes encapsulated and runs optimally on a very small, specific neural circuit without conscious input, what happens if the athlete suddenly starts consciously thinking about their mechanics again? Is this the neurological mechanism behind choking under pressure?
It absolutely is. Zhang's paper explicitly notes that once a motor skill reaches this high level of automaticity, directing conscious attention toward the execution of the movement actually degrades performance.
That is so counterintuitive.
When an athlete faces extreme anxiety or pressure, they often revert to step-by-step conscious monitoring. They try to forcefully control an action that their brain has already optimized to run subconsciously.
Like trying to manually control your breathing or blinking, it just messes it up.
By doing so, they disrupt the efficient, localized neural pathway and force the processing back into the widespread, slower novice networks. The movement becomes jerky, mistimed, and error-prone.
So the biological cure for choking is trusting the automated architecture.
Just let the subroutine run.
That is incredibly practical advice. Don't overrule your brain's optimization. But if the expert brain is operating on these highly localized streamlined pathways, we need to talk geography.
Let's do it.
Where exactly are these networks? If we take a map of the cortex, what specific regions distinguish the athlete from the amateur?
To map the precise anatomy of this expertise, we can look at a highly comprehensive study from 2025 by Wang and colleagues. It was published in the journal Frontiers in Sports and Active Living, titled Brain Networks of Athletes, a systematic review and meta-analysis of action anticipation and motor imagery.
Now, you mentioned earlier that a single fMRI study, like the one with the basketball and volleyball players, is powerful, but it might only have 20 or 30 participants per group due to the exorbitant cost of MRI time.
Right. Running those machines is incredibly expensive.
So, a meta-analysis aggregates data across multiple studies to find the undeniable biological consensus. But how do you aggregate fMRI brain scans? You can't just average the blood flow of 100 different people with different brain shapes.
It is a massive computational challenge. Wang and colleagues used 20 eligible fMRI studies and applied two sophisticated meta-analytic techniques.
Okay.
The first is ALE, which stands for Activation Likelihood Estimation, and the second is MACM, which is Meta-Analytic Connectivity Modeling.
Okay, those are some heavy acronyms. Let's break them down. What does ALE do?
ALE doesn't just average activation blobs. It treats every reported peak of activation from every study as a spatial probability distribution based on the sample size of that specific study.
So it weights the data based on how reliable the study was.
Exactly. It then calculates the statistical convergence of these probabilities across the entire brain. It essentially finds the geographical coordinates where activation is consistently likely to occur across all valid experiments, filtering out the statistical noise of small sample sizes.
So they are mathematically isolating the hotspots that consistently light up in athletes across different labs, different tasks, and different years.
You got it.
What did the ALE analysis reveal when they looked at athletes performing motor imagery?
The ALE analysis demonstrated that during motor imagery tasks, athletes consistently display significantly stronger activation in specific frontal lobe regions compared to non-athletes.
Okay, which regions?
The consensus hotspots were the left middle frontal gyrus, or the MFG, which structurally corresponds to Brodmann Area 8, and the left precentral gyrus, or the pre-CG, which is Brodmann Area 6.
Let's break down the functional roles of those specific Brodmann areas for the listener, because just throwing out anatomical names doesn't explain what the brain is actually doing. Why Brodmann Area 6 and Area 8?
Fair point. Brodmann Area 6 is located just in front of the primary motor cortex. It encompasses the premotor cortex and the supplementary motor area.
So it's involved in movement.
This is the command center for planning complex, coordinated, multi-joint movements. It stores the motor programs.
Okay, and Area 8?
Brodmann Area 8, the middle frontal gyrus, includes the frontal eye fields. This area is crucial for the management of uncertainty, the voluntary control of eye movements, and directing visual attention, tying beautifully back to our earlier discussion on multifocal tracking.
Oh, that makes perfect sense. So when athletes imagine a movement, they heavily recruit the premotor planning areas and the visual attention areas. But what about the other task they analyzed? Action anticipation. Like when an athlete is watching a video of an opponent moving and has to predict what they will do next.
This is where the data becomes truly fascinating. For action anticipation, the activation in athletes was even more robust in the motor planning areas.
Really?
Yeah, they showed massive, highly consistent activation in the left superior frontal gyrus, bilateral activation in the precentral gyrus, meaning both the left and right hemispheres of Brodmann Area 6 were heavily involved, and the right middle frontal gyrus.
So the athletes are heavily relying on the frontal motor planning areas to anticipate someone else's action. What were the non-athletes doing when they tried to anticipate actions in these same studies?
The non-athletes relied on entirely different cortical machinery.
Different machinery.
Yes. They showed greater activation in visual regions, specifically the left middle occipital gyrus, or MOG, which corresponds to Brodmann Area 17. That is the primary visual cortex located at the very back of the brain.
This is a profound distinction. The amateur is trying to predict the opponent's next move by using their visual cortex. They are treating it as a purely visual puzzle. They are just passively watching the movement.
Yes, exactly.
But the elite athlete is processing the opponent's movement using their own motor cortex.
Yes. To explain the mechanism behind this fundamental shift in processing, the Wang paper relies on the concept of the Action Observation Network, or the AON, and the mirror neuron system. They cite foundational work like Torrella's 2013 paper, Expertise in Action Observation.
Okay, the mirror neuron system, I've heard of that.
The Action Observation Network is a widespread circuit encompassing premotor and parietal areas. The defining feature of this network is that it activates both when you physically execute a specific action and when you simply observe someone else executing that same action.
I see. So an amateur watching a basketball game is sort of like a person watching a movie on a flat-screen monitor. They are taking in photons, processing shapes and colors in the visual cortex, and trying to logically deduce what is happening.
Right.
But the elite athlete watching that same game, it's more like they're wearing a high-end VR suit with full haptic feedback.
That is the perfect analogy.
Their brain isn't just passively seeing the opponent execute a crossover dribble.
Yep.
Their premotor cortex is physically simulating the neural commands required to do that crossover themselves.
Exactly. The athlete has spent thousands of hours physically performing and refining those exact kinematic movements. Consequently, their mirror neuron system is highly tuned, highly sensitized to the specific visual kinematics of that sport.
Wow.
When the athlete sees the opponent drop their shoulder by 2 degrees, the visual information instantly triggers the athlete's own motor programs for dropping a shoulder.
So they feel it.
Yes. The athlete's motor cortex simulates the physics, the balance, the necessary follow-through of that movement, allowing them to literally feel and predict the trajectory of the opponent fractions of a second before the opponent's movement is complete.
That is an astonishing evolutionary adaptation of the brain. We use our own motor system to empathize with and predict the physics of another human body.
It's incredible.
And the Wang paper didn't just find these isolated hotspots, did they? They used that second technique, MACM, to see how they wire together, right?
Yes, the meta-analytic connectivity modeling, or MACM, is crucial here. The brain doesn't work in isolated functional islands. MACM analyzes how different regions co-activate across studies to map functional connectivity.
What did that show?
The analysis demonstrated that in elite athletes, these specific regions form a highly cohesive, tightly integrated frontoparietal temporal network. This network seamlessly links the areas responsible for internally generated plans, the motor imagery, with the areas responsible for processing externally guided actions, the action anticipation.
So it's a super highway of processing.
It forms a highly specialized nested loop of high-speed processing.
And the amateurs in the studies, do they have that loop?
The researchers noted that non-athletes completely lack this cohesive functional network for sport-specific tasks. Their visual processing and motor planning regions remain largely functionally segregated when observing the sport.
We are talking about a fundamental structural rewiring of the cerebral cortex. The athlete is literally experiencing a different sensory and cognitive reality than the amateur when they step onto the court.
They absolutely are.
But to play devil's advocate for a moment, everything we have discussed so far, the visual tracking, the neural efficiency, the mirror neuron system, we have entirely been talking about the cerebral cortex, right? The wrinkled outer layer of the brain responsible for high-level cognition and sensory integration.
That is correct. The cortex manages the conscious strategy, the visual attention, and the high-level motor planning.
But basketball isn't played in a chair. It is an incredibly explosive, dynamic, gravity-defying sport. It requires absolute split-second biomechanical perfection while you are sprinting, jumping, and absorbing physical contact.
Very true.
To understand the raw subconscious engine of that kind of movement, looking at the cortex isn't enough. We have to look at the subcortical structures. We have to dive into the cerebellum.
We do. And historically, the cerebellum has been somewhat underappreciated in cognitive neuroscience.
Really? Why is that?
Well, it sits at the lower back of the brain, tucked underneath the occipital lobe. It contains more than half of all the neurons in the entire brain, despite only taking up about 10% of the total volume.
Half the neurons in 10% of the space. That's densely packed.
Incredibly dense. But for over a century, it was believed to be solely responsible for basic motor coordination, posture, and balance. We knew this largely from lesion studies. If you damage your cerebellum, you suffer from ataxia, meaning your movements become jerky, uncoordinated, and you lose your balance.
Right, a drunken stagger. But the scientific understanding of the cerebellum has evolved significantly, hasn't it? It's not just a dumb motor processor.
Far from it. Over the last few decades, highly advanced neuroimaging has revealed that the cerebellum is profoundly involved in higher-order cognitive functions, working memory, language processing, and the seamless integration of thought and movement.
So how does that relate to basketball?
To see exactly how the cerebellum adapts to the demands of athletic expertise, we have a groundbreaking new paper from 2025. It is titled Adaptations in the Structure and Function of the Cerebellum in Basketball Athletes, a multimodal MRI study.
Okay, multimodal MRI.
Authored by Key, Du, and their colleagues, and published in the journal Brain Sciences.
The title says a multimodal MRI study. This means they're doing much more than just looking at the boldy blood flow signal we talked about earlier. They're taking multiple different types of scans to build a comprehensive, three-dimensional profile of the athletic cerebellum.
Precisely. They wanted to capture the total architecture of the cerebellum, so they recruited a large cohort, 55 high-level elite basketball athletes, and matched them perfectly against a control group of 55 non-athletes.
Matched them how?
They matched them for age, education, and gender to eliminate any confounding developmental variables. They put all 110 subjects into the scanner and acquired 3 distinct types of data: macroscopic structural data, resting state functional data, and microstructural data.
Let's take these one by one, because this is where the biology gets incredibly granular. Let's start with the macroscopic structure. What specific technique did they use, and what did they find?
To analyze macroscopic structure, they used a technique called voxel-based morphometry to measure gray matter volume, or GMV.
Gray matter.
Right. Gray matter consists primarily of the neuronal cell bodies, the dendrites, and the synapses. It is the actual processing tissue of the brain as opposed to the white matter, which is the wiring connecting it.
So they're literally measuring the physical volume, the thickness of the brain tissue.
Yes. The VBM analysis revealed that the basketball athletes had significantly greater gray matter volume in a very specific bilateral region of the cerebellar cortex known as cruciferous. The physical tissue was measurably thicker and more voluminous than in the non-athletes.
Wait, does playing basketball actually grow physical volume in your brain? In
Well,
the same way that doing heavy bicep curls physically hypertrophies your muscle tissue?
In a functional sense, yes, though the biological mechanism is different. It is a phenomenon known as experience-dependent structural plasticity.
Experience-dependent structural plasticity.
Yeah, we aren't necessarily talking about neurogenesis, the birth of entirely new neurons, which is highly limited in the adult human brain. Rather, the structural expansion is driven by a massive increase in the density of synapses, the complex branching of dendrites to receive more signals, glial cell proliferation, and increased vascularization.
Just building more infrastructure to support the demand.
Exactly. Increased vascularization to support the heightened metabolic demand of that specific region. The gray matter physically expands in response to the chronic computational demands placed upon it.
Incredible. Okay, so the hardware is physically larger in cruciferous. What about the second measure? Resting state function. How do you measure the function of a brain that is just resting?
The brain is never truly silent. Even when you are lying perfectly still in a scanner with your eyes closed, not performing any task, there are slow, spontaneous fluctuations in the bold D signal.
Just background noise.
Analyzing these resting state fluctuations provides a map of the brain's baseline intrinsic functional architecture. Key and colleagues looked at 2 specific resting state metrics: ALFF, which stands for amplitude of low-frequency fluctuations, and ReHo, which stands for regional homogeneity.
What is the difference between ALFF and ReHo? What do they tell us about the athlete's brain?
ALFF measures the raw intensity or amplitude of those spontaneous fluctuations within a specific voxel. It is generally interpreted as a measure of the baseline neural energy or the intrinsic functional readiness of that region.
And the athletes.
The athletes showed significantly increased ALFF in the right cruci.
So their cruci is not just physically booger, it's idling at a higher RPM. It has a higher baseline level of readiness.
Precisely. Now, ReHo, on the other hand, measures local synchronization. It calculates how similar the time series of a specific voxel's activity is to the activity of its immediate neighboring voxels.
Like a choir singing in perfect harmony versus everyone singing their own song.
That's a great way to think of it. A higher ReHo indicates highly synchronized, cohesive local neural firing. The athletes showed significantly higher ReHo in areas of the cerebellum known as crus II and lobule VII.
So the processing tissue is bigger, it has a higher energetic baseline, and it fires with greater local synchrony. Now, what was the third measure? The microstructure. This sounds like we were zooming in even further.
This is the most technically complex, but arguably the most fascinating measure. They use an advanced technique called diffusion kurtosis imaging, or DKI.
Okay, tell me about DKI.
Standard diffusion tensor imaging, or DTI, tracks how water molecules diffuse through the brain tissue to map the white matter tracks. In a completely open environment, water diffuses isotropically equally in all directions, like a drop of ink in a glass of water.
Sure.
But in the brain, the diffusion of water is restricted by the microscopic cellular architecture. The rigid cell membranes, the myelin sheaths wrapped tightly around axons, the densely packed microtubules. Water is forced to diffuse anisotropically, moving mostly along the length of the neural fiber tracks.
So by tracking how the water is restricted, you can indirectly map the structural integrity of the brain's internal wiring, the fiber optic cables connecting the processing hubs. But what makes diffusion kurtosis imaging different than standard diffusion imaging?
Standard diffusion imaging assumes the water diffusion follows a normal Gaussian distribution, but biological tissue is incredibly complex and heterogeneous. DKI specifically measures the non-Gaussian diffusion of water.
The kurtosis.
Yes, it measures the kurtosis, the degree to which the diffusion is restricted by incredibly complex microenvironments, like crossing fibers or densely packed cellular compartments. They focused on two DKI parameters: kurtosis fractional anisotropy, or KFA, and radial kurtosis, or RK.
Without getting completely lost in the fluid dynamics, what do changes in KFA and RK actually indicate about the athlete's brain wiring?
Generally speaking, in this context, an increase in KFA and a decrease in RK indicate a higher degree of microstructural complexity, more tightly ordered and densely packed neural fiber structures, and potentially thicker myelination.
So better, faster cables.
It indicates a highly optimized, high-fidelity biological wiring system.
And the results for the athletes.
The DKI analysis showed significantly increased KFA and decreased RK throughout the cerebellar cortex and its peduncles,
Peduncles.
which are the massive bundles of nerve fibers connecting the cerebellum to the brainstem and the rest of the cortex. And crucially, these microstructural modifications were, once again, primarily localized around Cara's and lobule vive.
Okay, this is remarkable. We have 3 entirely different types of MRI data measuring physical volume, baseline functional energy, and microscopic water diffusion.
Yeah.
And all 3 point unequivocally to the exact same geographical real estate in the cerebellum, Crisai.
Yes, Crisai.
Why is Crisai the biological holy grail for a basketball player?
To understand that, we have to look at the anatomical connectivity of Crisai. As we discussed, the cerebellum is not just a motor organ. Crisai is a critical node in cerebellar cognitive loops. It has massive reciprocal connections with the prefrontal cortex, the area governing executive function, decision-making, and working memory, and the parietal cortex.
Wait, the exact same cortical areas we were talking about earlier. The areas responsible for anticipating actions and spatial awareness.
Exactly. They are fundamentally linked. Consider the demands of basketball. An elite player must make split-second tactical decisions, recognizing a complex defensive rotation, calculating the trajectory of a pass, deciding whether to shoot or drive.
These are high-level cognitive tasks.
Right. But they must seamlessly integrate those cognitive decisions with exact, extraordinarily complex biomechanical execution, like adjusting the release point of a jump shot while drifting laterally in midair to avoid a defender.
Right, cognitive thought turned into physical reality.
The structural expansion and the enhanced microstructural wiring of Crisai provides a faster, more robust computational internal model for the athlete. It is the integration hub that allows thought to become flawless movement without any delay.
Did Key and his team actually prove that these brain modifications translate to physical dominance on the court? Because it's one thing to say they have a bigger Crisai, it's another to prove that makes them jump higher.
They absolutely proved the behavioral correlation. They took the athlete's physical fitness test scores and ran correlational analyses against the brain metrics.
Okay, what did they find?
They found that KFA, that measure of dense, efficient microstructural wiring in the cerebellum, positively correlated with the athlete's vertical jump score, yielding an effect size of R equals 0.45, with a statistically significant p-value of 0.021.
Okay.
Furthermore, the ALFF, the baseline resting energy in Croces, I also correlated with the vertical jump, with an effect size of R equals 0.52.
For our listeners who might not look at statistical outputs every day, how should we interpret an R value of 0.45 or 0.52 in this context?
In human behavioral neuroscience, where data is incredibly noisy because humans are incredibly complex, an R value around 0.5 is considered a very strong correlation.
Wow.
It means that a highly significant portion of the variance in why one elite athlete can jump higher than another elite athlete is directly, linearly linked to the microstructural efficiency and the functional readiness of their cerebellum. The brain architecture predicts the physical output.
The hardware dictates the performance.
Exactly. Additionally, they found that RIHO, the regional synchronization, correlated with their total years of basketball training with an R value of 0.31. The longer they subjected their brain to the demands of the sport, the more synchronized those specific cerebellar regions became.
We are looking at the biological blueprint of athletic genius. We've established that the brain undergoes massive structural plasticity, cortical rewiring, and subcortical expansion.
We have.
But this begs the ultimate billion-dollar practical question for coaches, athletes, and anyone trying to master a complex skill.
How do we get there?
Exactly. If the brain can typically change this much, how do we optimally trigger that neuroplasticity?
Yeah.
Because it seems like just doing standard practice, just standing on an empty court, shooting 1,000 free throws, isn't enough.
No, it's not.
It builds the motor subroutine, but does it build the cognitive motor integration required in a real game? How do we specifically target these advanced neural pathways?
That is the frontier of sports training. Standard repetitive motor drilling has severe limitations because it doesn't challenge the prefrontal cerebellar cognitive loops we just discussed.
So what does?
To see how we actually build the expert brain, we are going to examine a fascinating clinical trial from 2021 titled Effects of a Cognitive Motor Training on Anticipatory Brain Functions and Sport Performance in Semi-Elite Basketball Players. This was authored by Lucia and a team of researchers and published in the journal Brain Sciences.
I noticed the title specifies semi-elite players. That is a crucial detail. If you take absolute beginners and give them any type of training, they will improve rapidly. It's easy to show an effect size with novices.
Exactly. The newbie gains.
But semi-elite players are already highly optimized. It is incredibly difficult to move the needle on performance when someone has already been playing for 10 years. How did Lucia design the experiment to break through that plateau?
The experimental design was highly controlled. They recruited 24 young, semi-professional basketball players and randomly divided them into two groups of 12. The intervention lasted for 5 weeks.
5 weeks, okay.
Now, during this time, both groups maintained their normal, mandatory team practices and matches. The variable was introduced in 2 extra individual training sessions per week. The control group participated in standard motor training. These were intense, high-level, physical and technical basketball drills they were already accustomed to.
And the experimental group, what did they do differently?
The experimental group underwent what is called cognitive motor training, or CMT. In these sessions, the athletes were required to perform complex physical dribbling exercises, but simultaneously they were subjected to a heavy cognitive load.
A heavy cognitive load, like what?
They used interactive electronic devices, specifically specialized light pods that were arranged around them and emitted unpredictable visual and auditory stimuli.
So they're dribbling 2 basketballs, doing crossovers, and suddenly a pod to their left flashes blue and a pod to their right beeps.
Exactly. And they had to process these stimuli, apply a complex set of predetermined cognitive rules, for example. If the pod flashes blue, touch it with your left hand, but if it flashes red and beeps, change your dribble direction and physically execute the correct motor response while maintaining absolute control of the basketball.
That sounds neurologically exhausting.
It is brutal.
It is the ultimate test of divided attention. You are demanding absolute biomechanical perfection from the motor cortex while forcing the prefrontal cortex to solve logic puzzles in real time. How did the researchers measure the neurological impact of this training?
Both before the 5-week intervention began and after it concluded, the researchers subjected all the athletes to a go-no-go discrimination response task, or DRT. And crucially, while performing this cognitive task, the athletes were hooked up to an EEG, an electroencephalogram, to record their brainwave activity in real time.
Let's quickly differentiate EEG from the fMRI we discussed earlier.
Yeah.
fMRI gives us beautiful, high-resolution spatial maps of where blood is flowing, but it's slow. EEG doesn't give you great spatial resolution, but it measures the actual electrical firing of the neurons with millisecond precision, right? It gives you the exact temporal dynamics of thought.
That is precisely why they used it. They wanted to see the millisecond timing of anticipation. In the go-no-go task, the athlete sits at a computer and is presented with a rapid, unpredictable series of visual stimuli.
Okay, what do they have to do?
They are given a strict rule. Respond as quickly as humanly possible by pressing a button when you see a specific go target, but you must actively inhibit your response. You must do absolutely nothing when you see a slightly different no-go distractor.
Which perfectly mimics the cognitive demands of basketball. A point guard initiates the motor sequence to throw past the go state, but at the last possible millisecond, They see a defender flash into the passing lane, and they have to aggressively abort the motor command, the no-go state. What specific brain waves was the EEG looking for during this process?
The EEG allowed researchers to extract and analyze event-related potentials, or ERPs. ERPs are the measured brain responses that are the direct result of a specific sensory, cognitive, or motor event. They specifically zeroed in on two ERP components that occur approximately 1 full second before the visual stimulus even appears on the screen.
A full second before.
Yes, these are anticipatory waves. The first is the Bereitschafts potential, or BP, which originates over the motor areas and indicates subconscious motor preparation.
The readiness potential.
Exactly. The second is the prefrontal negativity, or PN, which originates over the prefrontal cortex and represents cognitive anticipation and the allocation of top-down attention.
They are measuring the brain, revving its engine in anticipation before the green light even flashes. What were the findings after the 5 weeks of cognitive motor training? Let's start with the behavioral physical results.
The behavioral results were profound. Post-treatment, the experimental CMT group showed significant improvements in all their sport-specific physical tests, improving overall by roughly 17%.
17%?
Yeah. For semi-elite athletes, a 17% jump in physical execution over just 5 weeks is an enormous effect size. The control group showed marginal, non-significant improvements.
And what about their performance on the cognitive go-no-go task?
This is where the data is truly stark. On the go-no-go task, the standard motor training control group achieved an overall response accuracy of about 60%. The cognitive motor training group hit an astonishing 88% response accuracy.
88% versus 60%. That is a massive discrepancy in cognitive control and proactive inhibition. Did the EEG data explain the biological mechanism behind that jump?
It did, beautifully. At the neurological level, the ERP analysis showed that in the experimental group, the anticipatory processes, those BP and PN waves, began significantly earlier in time.
So they were faster.
And the amplitude of the waves was significantly heightened compared to the control group. The training had physically altered their brain dynamics, making their prefrontal cortex vastly faster and more powerful at allocating top-down attention and preparing proactive inhibition before an event even occurred.
So the takeaway for anyone listening is clear. If you are trying to master a complex physical skill, whether that is basketball, playing a complicated piece on the piano, or even learning a highly technical surgical procedure, do not just drill the physical motion on autopilot.
Never on autopilot.
If you just do reps, you are only building the subcortical motor loop. You have to actively add a heavy cognitive load. You have to force yourself to solve novel problems, make decisions, or react to unpredictable stimuli while executing the movement.
Exactly. You must force the cognitive networks and the motor networks to fire simultaneously under high stress. And to truly understand how this works computationally at the synaptic level, we can look at an incredibly influential paper published in 2006 by Paul Cisek.
What's the paper?
It's titled Integrated Neural Processes for Defining Potential Actions and Deciding Between Them, a computational model.
A computational model?
Yeah.
So Cisek didn't put people in a scanner. He built a mathematical simulation of the brain to prove how these networks interact.
Yes, Cisek modeled large populations of what he defined mathematically as mean-rate leaky integrator neurons.
Okay, I have to stop you there. We cannot gloss over mean-rate leaky integrator neurons.
Yeah.
We promised to dive deep into the science. What does that actually mean in the context of a biological brain cell?
Right. It is a mathematical model that perfectly mimics biological reality. In your brain, a single neuron doesn't just instantly fire when it receives a tiny signal.
It's not just an on-off switch.
No, it receives electrical inputs from thousands of dendrites. It integrates or sums up these electrical inputs over time. However, the neuron is leaky. The electrical charge naturally dissipates or leaks out through the cell membrane. Therefore, in order for the neuron to reach the critical threshold and fire an action potential, the incoming signals must be strong enough and frequent enough to overcome the constant leakage.
Yes.
The mean rate aspect simply means the model looks at the average firing rate of a population of these neurons rather than tracking every single spike.
So a leaky integrator neuron is basically like a bucket with a small hole in the bottom. If you drip water in slowly, it just leaks out and the bucket stays empty. But if you pour water in rapidly from multiple sources, the bucket fills up, overcomes the leak, and eventually overflows, which is the neuron firing.
That is an excellent visualization. Cisek used vast networks of these modeled neurons to mathematically simulate how different regions of the brain interact during complex decision-making.
Which regions?
Specifically, his model demonstrated how the prefrontal cortex, the PFC, which handles cognitive rules, goals, and logic, interacts with the dorsal premotor cortex, the PMD, which handles the spatial preparation of the actual motor actions based on visual input.
How do they interact? Does the prefrontal cortex just command the motor cortex what to do?
It is more nuanced than a direct command. Cisek's model shows that the prefrontal cortex provides a low-resolution bias signal that multiplicatively modulates the activity in the dorsal premotor cortex.
Let's translate multiplicatively modulates into plain English.
Think of the dorsal premotor cortex as a massive audio mixing board with various sliders representing different possible physical movements you could make right now.
Okay, a mixing board.
The raw visual input from your eyes pushes several of these sliders up simultaneously, preparing multiple potential actions based on the spatial environment.
So your brain is preparing several options at once.
Exactly, but the prefrontal cortex acts like a master volume dial for specific channels. It doesn't initiate the movement itself. Rather, it multiplies the neural firing rate of the correct movement plan.
Rapidly pushing that specific leaky bucket past the threshold to fire.
Yes.
Yeah.
While actively suppressing the firing rates of the incorrect movement plans, it is a multiplicative gating mechanism.
That brings it all together perfectly.
Mm.
When the athletes were doing the cognitive motor training, when they were forced to solve the light pod puzzles using the logic rules in their prefrontal cortex, while simultaneously maintaining their complex dribbling using their premotor cortex, they were forcing those two regions to fire at maximum capacity at the exact same time.
They absolutely were.
As the old neuroscience adage goes, neurons that fire together wire together.
Precisely. Standard motor training only exercises the isolated premotor and cerebellar motor loops. The cognitive gating mechanism remains largely unchallenged.
But the CMT training changes that.
Yes, cognitive motor training forces the prefrontal cortex to physically strengthen its synaptic connections, its multiplicative gating pathways deeply into the motor execution networks,
It literally thickens the wiring between thought and action.
granting the athlete vastly superior, instantaneous conscious control over rapid, automatic movements.
This has been an absolutely staggering journey through the architecture of the expert brain. Let's take a moment to briefly synthesize everything we've unpacked today.
It's a lot of ground to cover.
It really is. We started at the perceptual level, exploring how athletes visually track the chaotic world around them. We learned that human attention is strictly divided between our visual hemifields, and that through intense training, experts develop high-resolution multifocal tracking,
Allowing them to shrink their zones of interaction and process multiple targets simultaneously without cognitive interference.
Exactly.
Yeah.
Building on that perceptual foundation, we moved to the neural efficiency hypothesis. We discovered the counterintuitive reality that true mastery doesn't mean the brain is working harder.
Instead, expert brains undergo massive synaptic pruning, allowing them to use less oxygen and rely on highly localized, incredibly streamlined automated pathways.
Which protects them from the performance-destroying effects of conscious overthinking or choking.
Right.
We then mapped the physical rewiring of the cerebral cortex, revealing how athletes uniquely utilize the action observation network and the mirror neuron system.
By relying heavily on premotor planning areas like Brodmann Area 6, they physically simulate the movements of their opponents, shifting their processing from reactive visual seeing to predictive haptic feeling.
Incredible. And from the cortex, we descended into the subcortical engine of movement, the cerebellum. We analyzed multimodal MRI data, proving that long-term athletic training physically increases gray matter volume and optimizes the microstructural fiber wiring, specifically in cerebellar cruci.
And we saw how the structural integrity of this cognitive motor integration hub linearly correlates to physical outputs like vertical jump height.
And finally, we explore the frontier of sports science with cognitive motor training, utilizing Cisek's computational models to understand how applying heavy cognitive loads during physical drills forces the prefrontal cortex to wire deeply into the premotor cortex.
Massively enhancing an athlete's anticipatory cognition and their ability to proactively inhibit incorrect actions.
It was a remarkable testament to the power of human neuroplasticity.
Yeah.
But before we sign off, there is a final, highly provocative implication hiding within the data of these source materials that we haven't fully explored yet.
Oh, there certainly is.
I am always ready for a provocative thought to end on. Where does this data logically lead us?
Well, if we look back at Yarrow's 2009 discussion regarding predicted physiological profiling, and we synthesize it with Qi's brand new 2025 findings on highly specific cerebellar biomarkers like the kurtosis fractional anisotropy and radial kurtosis metrics in cresolite,
Okay, I'm tracking.
it raises an astonishing possibility for the future of athletics. If we know that the cerebellum adapts so distinctly to the integrated cognitive motor demands of a specific sport like basketball, could we eventually use multimodal MRI to scan a 10-year-old child's baseline cerebellar microstructure, analyze their intrinsic functional connectivity, and predict with scientific certainty the exact sport their brain is biologically optimized to master?
That is heavy. Biological destiny dictated by an MRI scan. We could stop guessing what sports kids might be good at and just read their cerebellar blueprints.
Or, taking it a step further into the realm of science fiction, though a realm that is entirely grounded in the mechanics of neuroplasticity we've discussed today.
Go on.
If we know the exact structural changes and network cohesions in brain regions like cresci and the precentral gyrus that dictate expertise, will the future of elite sports training involve using targeted, non-invasive brain stimulation?
Wait, like zapping the brain.
Could we eventually use transcranial magnetic stimulation or direct current stimulation to artificially induce these microstructural changes? Could we effectively download the neurological architecture of expertise into an athlete's brain, bypassing or at least massively accelerating what currently requires thousands of hours of deliberate practice?
That is a staggering thought to leave everyone with. The idea that the biological gap between the amateur and the master, built through decades of sweat and repetition, might one day be bridged by directly stimulating the neural circuitry we have spent the last hour mapping out.
It changes everything we know about human potential.
It really does. So the next time you are sitting in an arena and you see that point guard execute that impossible, chaotic, flawless, no-look pass, remember that you aren't just looking at a physical marvel of muscle and bone.
No, you definitely are not.
You are watching a finely tuned, hyper-efficient, structurally modified neural supercomputer operating at the absolute breathtaking limits of human biology.
Beautifully said.
Thank you so much for joining us on this deep dive. We hope you take these insights into multifocal tracking, neural efficiency, and cognitive motor integration, and apply them to your own pursuit of mastery, whatever your chosen field may be. Until next time, keep learning, keep questioning, and keep diving deep.